Alright, hello doctors, welcome back to Paratha Medicine. Today we are back with the biochemistry based solution series. The last video in this series, today we are going to give it to you, and I hope this series helped you in your Krok preparation, and I also hope that you are on track for your Krok exam on 29th of June or 30th of June, whatever date is it. So without any further ado, let's end this. 146 is that a sportsman needs to improve his sporting results. He was recommended to take a preparation that contains carnitine. What process is activated the most by this compound? Now we have done this topic again and again. So carnitine is involved in fatty acid oxidation. How? It transports fatty acids into mitochondria. So the first step is activation of fatty acids into acyl coenzyme A. Now carnitine acts as a carnitine shuttle and carries acyl coenzyme A into the mitochondria because the mitochondrial membrane is impermeable to fatty acid as its own. So what we do? Carnitine combines with acyl coenzyme A to form acyl carnitine, goes in and gives off acyl coenzyme A and returns back to the cytosol and continues this shuttle again and again. So this is how carnitine helps. It is involved in fatty acid transporting. 147. A doctor examined a child and revealed symptoms of rachitis or rickets. Development of this disease was caused by the deficiency of the following compound. So if you have any medical knowledge, you know that rickets is caused by the deficiency of vitamin D, and the active form of vitamin D is 1.25 dihydrocholecalciferol. If you remember, we studied about this, that in the kidney, the 25 hydroxycholecalciferol is converted into 1.25 dihydroxycholecalciferol or calcitrol, the active form of vitamin D, correct, with the help of enzyme 1-hydroxylase. So this calcitrol or 1,2,5 dihydroxycholecalciferol is the active form of vitamin D, and vitamin D is deficient in rickets. So that is why the answer is 1.25 dihydroxycholecalciferol. 148. Lab examination of a child revealed increased concentration of leucine, valine, isoleucine and their keto derivatives in the blood and urine. Note, urine smelt of maple syrup. This disease is characterized by the deficit of the following enzyme. So if you remember, we talked about this also, that we have these branched chain amino acids, valine, leucine and isoleucine, and they have this characteristic disorder in their metabolism, metabolic disorder, maple syrup urine disease. Now if you understand the branched chain amino acids metabolism, you will see that valine, leucine and isoleucine with the help of enzyme branched chain amino acid transaminase are converted into corresponding alpha keto acids. Now for example, the valine's alpha keto acid is alpha keto isovalerate, leucine is alpha keto isocaporate, and isoleucine is alpha keto beta methyl valerate. Now these keto acids are further converted into corresponding alpha beta unsaturated acyl coenzyme A thioesters with the help of the enzyme alpha keto acid dehydrogenase complex. Now if you remember, maple syrup urine disease is a disorder of this enzyme complex, alpha keto acid dehydrogenase complex. So if there is an enzyme defect of this enzyme, what would happen? Corresponding keto acids and the branched chain amino acid will accumulate in the blood, which will cause the maple syrup urine disease. The accumulation of these keto acids and the branched amino acids in the plasma and urine causes this characteristic smell of maple syrup or burnt sugar, and that is why this is called as maple syrup urine disease. I hope you understand the biochemistry behind maple syrup urine disease. So 149. A 9-month-old child infant is fed with the artificial formula with the balanced vitamin B6 concentration. The infant presents with pellagra dermatitis, convulsions, anemia. Now they are asking, convulsion development might be caused by the disturbed formation of what? Now if you remember, glutamine is converted into glutamate by the help of glutamine synthetase. Now this glutamate is converted into GABA, the inhibitory neurotransmitter of our body, by the enzyme glutamate decarboxylase. And the coenzyme which takes part in this reaction, coenzyme of glutamate decarboxylase, is vitamin B6 or pyridoxal phosphate. So if this vitamin B6 is deficient, then glutamate decarboxylase will not work, which will cause increased accumulation of glutamate and hence convulsions. So that is why the answer here is convulsion development might be caused by the disturbed formation of GABA. Correct? So there we go. Vitamin B6 deficiency, depression, irritability and mental confusion. Convulsions are absorbed in severe deficiency. These are related to the decreased synthesis of GABA. Correct? So some additional information, you can note it down, that in children specifically, B6 deficiency leads to reduced GABA production, resulting in convulsions and epilepsy, and also decrease in hemoglobin levels associated with hypochromic microcytic anemia is seen in B6 deficiency, which results in reduction in heme production, because pyridoxal phosphate, vitamin B6, is also important coenzyme in heme synthesis. Anyway, let's go to the next one. 150, it was found out that some compounds, for instance fungi, toxins and some antibiotics, can inhibit the activity of RNA polymerase. What process will be disturbed in a cell in case of inhibition of this enzyme? Now if you remember RNA polymerase is an important enzyme that takes part in the process of transcription, formation of mRNA from a DNA. So this is the RNA polymerase complex. What it does, it moves throughout the unwounded DNA and generates complementary mRNA, and that is the process of transcription. Correct? So simple. Let's go to the 151 question. Then a blood circulation in a damaged tissue is restored. Then lactate accumulation comes to a stop and glucose consumption decelerates. These metabolic changes are caused by the activation of the following process. Now, we talked about anaerobic glycolysis, we talked about aerobic glycolysis. What happens in anaerobic glycolysis is that in case of absence of oxygen, glucose is converted by lactic fermentation into lactate, because it is a faster mechanism of providing ATP and it does not involve use of oxygen. So in case of a short supply of oxygen, when the body is not able to provide enough oxygen, what happens is that the muscle uses lactate method to make ATP, but this leads to accumulation of lactate. Now this is the thing that happens in deficiency of oxygen, in excessive muscle work. Now they said that increased lactate accumulation comes to a stop when blood circulation is restored. So when anaerobic glycolysis comes to a stop, what does it begin? Aerobic glycolysis, the normal process. After the abnormal process or the compensatory process has stopped, the normal process of aerobic glycolysis will continue. So this is caused by the activation of aerobic glycolysis. Just don't go for anaerobic glycolysis. If you see lactate written in the question, don't abruptly go to anaerobic glycolysis. Remember to read the question and understand what they are asking in the question. So this is a classical example of, if you just underline something randomly and remember, oh, lactate means anaerobic glycolysis, you're going to get it wrong. So let's go to the next one. 152. During starvation, muscle proteins break up into free amino acids. These compounds will be the most probably involved in the following process. So in starvation, what happens? Your glucose reserve is depleted, and then when muscle proteins are breaking up, which means that fat stored is also depleted, and this is the third stage in which the proteins are going to be used for making energy. So for making energy, what happens is that these various first of all, the proteins break into their corresponding different amino acids. Now these amino acids, they enter the process of gluconeogenesis, so that they make glucose, and the glucose go into the process of TCA cycle, so that energy is being able to produced, and so that you can use this energy for your survival. And this is the various places where various amino acids get accumulated or get introduced into the TCA cycle. So you can see various amino acids like tryptophan, tyrosine, phenylalanine, threonine, isoleucine, lysine and leucine can directly convert into acetyl coenzyme A and carry on with the process of gluconeogenesis. Alanine, glycine, cysteine, serine, threonine and tryptophan. These amino acids can also form pyruvate. Then arginine, histidine, proline, glutamine form glutamate first of all, and this glutamate is converted into alpha ketoglutarate. Then threonine, isoleucine, methionine, valine form succinyl coenzyme A. Threonine, tyrosine, phenylalanine and aspartate, they form fumarate. Now the thing to remember is that amino acids can be glucogenic or can be ketogenic. By glucogenic it means that these amino acids, they form a glucose molecule first of all. So for example, all these glucogenic, they get introduced into the TCA cycle directly, or a glucose metabolite. And see, you can see that arginine, glutamine, all these given in red, glucogenic, they are introduced into the TCA cycle. So these are glucogenic. Then we have ketogenic. Now ketogenic are specific for forming acetyl coenzyme A. Now from acetyl coenzyme A, they can form ketone bodies, but here they are getting used for gluconeogenesis. So ketogenic amino acids, they form ketone through acetyl coenzyme A, while there are some both glucogenic and ketogenic given in purple. So remember, we have lysine and leucine as ketogenic amino acids, and remember the red ones as glucogenic, and all other remaining are both glucogenic and ketogenic. So I hope you understand how these amino acids, how this protein breakdown is involved in the process of gluconeogenesis in the liver. Remember, gluconeogenesis occurs in the liver, not in the muscles, not anywhere else. It happens in the liver, and that is why in starvation proteins break into amino acids, and these amino acids are being used for gluconeogenesis in liver. 153. Surgical removal of a part of stomach resulted in the disturbed absorption of vitamin B12. It is now excreted with feces. The patient was diagnosed with anemia. What factor is necessary for absorption of the vitamin B12? So vitamin B12 absorption occurs with the help of a special factor called as Cassell's intrinsic factor. Now this Cassell's intrinsic factor is secreted by the gastric parietal cells in the gastric mucosa. Now this intrinsic factor, when it is secreted in the gastric fluid, it travels through the gut and reaches the ileum part of the small intestine. In the ileum, it binds with a specific receptor on the surface of ileum, and then it combines first of all with the vitamin B12 in the gastric mucosa. So this intrinsic factor and vitamin B12 complex travels to the ileum and binds to a surface of ileum's mucosa and goes into the cell with the help of an active process. So ATP is being used here. This is how vitamin B12 enters into the blood, and this protein and forms a plasma protein which is called as transcobalamin, which carries the cobalamin to liver and various tissues. So remember, for vitamin B12 absorption, Cassell's intrinsic factor is responsible and very much needed. And deficiency of Cassell's intrinsic factor can cause anemia, because vitamin B12 is necessary for production of hemoglobin. And remember, Cassell's intrinsic factor is a type of gastromucoprotein. So that is why the answer is gastromucoprotein. 154. A newborn develops dyspepsia after the milk feeding. When the milk is substituted by the glucose solution, the dyspepsia symptoms disappear. The newborn has the subnormal activity of the following enzyme. Note that the baby develops dyspepsia after milk feeding. So the baby obviously is only taking milk, and milk contains sugars and proteins. And we have these conditions called as lactose intolerance. So some people are lactose tolerant and some people are lactose intolerant. In lactose intolerant people, we have this enzyme called as lactase. Now this lactase is responsible for converting the lactose sugar present in milk into, lactose is a disaccharide. It is made up of two sugars. Now lactase is responsible for breaking the bond between the two sugars and forming monosaccharides, the glucose monosaccharide and the galactose monosaccharide. So in people who are lactose intolerant, they don't have lactase. So what happens? Lactose is not converted into glucose and galactose. Now because it is not converted, then it cannot be absorbed. It goes to the large intestine, and the bacteria present in large intestine use this lactose to form various acids and gas, which causes the problem of flatulence and abdominal pain, which is characteristic for lactose intolerant people. So that is why the people who are lactose intolerant have a subnormal activity of lactase. 155. A patient who suffers from severe diabetes and doesn't receive insulin has metabolic acidosis. This is caused by the increased concentration of, we already know that the people who suffer from severe diabetes, we talked about this in physiology. So people who suffer from severe diabetes, they suffer from a condition in extreme complications called as diabetic ketoacidosis. In this case, what happens is there is an increased production of ketone bodies in the body. Now these ketone bodies are acidic in nature, and their increased concentration causes metabolic acidosis. Correct? I hope you understand this. So let's go to the next one. 156 is that a 4-year-old child with hereditary renal lesion has signs of rickets. Vitamin D concentration in the blood is normal. What is the most probable cause of rickets development? Now the thing to note here is that the vitamin D concentration is normal. What they mean from this is that vitamin D, the 7-dehydrocholecalciferol, is normal in the body. But you need to know that vitamin D is metabolized in the body into its active state, which I already told you. 7-dehydrocholecalciferol is converted into cholecalciferol with the help of UV light, and this cholecalciferol goes to the liver and it is converted into 25-hydroxycholecalciferol in the liver with the help of enzyme 25-hydroxylase. It goes to the kidney. In the kidney, 1-hydroxylase enzyme converts it into 1,2,5-dihydroxycholecalciferol, calcitrol, the active form of vitamin D. And this vitamin D's active form is responsible for all the metabolic functions of the vitamin D, like increasing calcium and phosphate absorption in the kidney, going to the bone and increasing the bone turnover and formation ratio, going to the intestine and increasing the absorption of calcium. So this responsible function of the vitamin D is by 1,2,5-dihydroxycholecalciferol, also called as calcitrol. So even though person has a vitamin D normal concentration, but here it is a disorder of impaired synthesis of calcitrol in the kidneys. So that is why even though vitamin D concentration is normal, this child suffers from rickets. 157 is a 6-year-old child was delivered to a hospital. Examination revealed that the child couldn't fix his eyes, didn't keep his eyes on toys, eye ground had a cherry red spot sign. Lab analysis showed that the brain, liver and spleen had high rate of ganglioside glycolypid. What congenital disease is the child ill with? Now remember, we have various storage diseases of gangliosides and cerebrocytes. Okay, so we have the Niemann-Pick disease, in which the defective enzyme or the missing enzyme is sphingomyelinase, and major storage compound is sphingomyelin. So there are symptoms of enlargement of liver, spleen, mental retardation. Then we have Farber's disease, in which ceramidase is absent and ceramide is the major storage compound. Gaucher disease, beta glucosidase, glucocerebroside is stored and this enzyme is deficient, beta glucosidase. And enlargement of liver, spleen, osteoporosis, mental retardation are the symptoms. Then we have Krabs disease, in which the enzyme beta galactosidase is deficient, galactocerebrosides are accumulated, and it leads to absence of myelin formation, liver and spleen enlargement, mental retardation again. And we have the Tay Sachs disease, in which the enzyme deficient is hexoaminidase A, and the accumulated compound is ganglioside glycolypid 2. It leads to symptoms like blindness, mental retardation, death within 2 years. Some simple symptoms here given. We'll talk about them in the next slide. Then we have the Fabry's disease, in which the enzyme deficient is alpha galactosidase. Again, here was beta galactosidase, here is alpha galactosidase. Accumulated compound is ceramide trioxide. Renal failure, skin rash, pain in the lower extremities are the symptoms. Now, characteristic symptoms of the Tay Sachs disease, this one, are there is a loss of learned skills, loss of smile, growl and grabbing of the babies, blindness, deafness, paralysis, dementia, unable to swallow, muscle atrophy, and a characteristic cherry red spot on the retina on the back of their eyes. This is seen. This cherry red spot is due to degeneration of the optic nerve. Correct? Because of this enzyme not working and accumulation of ganglioside glycometide. So this is seen in Tay Sachs disease. 158. In a clinical practice, tuberculosis is treated with the Isoniazid preparation. That is an antivitamin able to penetrate into the tuberculous bacillus. Tuberculostatic effect is induced by the interference with the replication processes and oxidation reduction reactions due to the buildup of a pseudo coenzyme. Now what happens is that Isoniazid goes into the Mycobacterium tuberculosis and it does various reactions. It is converted into isonicotinic acid by the enzyme CATG. Now CATG is the enzyme in the bacteria that activates Isoniazid drug. This also leads to formation of various reactive organic and oxygen radicals which damage the DNA of the Mycobacterium. They inhibit the mycolic acid synthesis, and this Isoniazid forms a NAD adduct pseudo coenzyme in the Mycobacterium tuberculosis, which again inhibits mycolic acid, and if you remember, it leads to the death of Mycobacterium tuberculosis. Now remember, they ask you in one single line, how does Isoniazid show its mechanism of action? By inhibiting mycolic acid synthesis in the bacteria. Now, mycolic acid is an important component of this bacteria's cell wall, Mycobacterium tuberculosis cell wall. Because it is not synthesized properly, this leads to the death of the bacteria by the action of Isoniazid. So the coenzyme, pseudo coenzyme formed here is NAD, nicotinamide adenine dinucleotide, NAD. Correct? Good. So 159. A newborn child was found to have reduced intensity of sucking, frequent vomiting, hypotonia. Now urine and blood exhibit increased concentration of citrulline. What metabolic process is disturbed? Let's talk about urea, ornithine, citrulline cycle and the disorders related to it. If you remember, in the liver, carbon dioxide and ammonia, they convert into carbamyl phosphate with the help of enzyme carbamyl phosphate synthase 1. Remember, not 2. 2 is involved in pyrimidine synthesis, not 2. So here 1 is involved. This carbamyl phosphate combines with ornithine to form citrulline with the help of enzyme ornithine transcobimylase. Then the citrulline combines with aspartate to form arginosuccinate with the help of arginosuccinate synthase. Arginosuccinate gives fumarate out, and arginosuccinase enzyme acts during this process to form arginine amino acid. This arginine hydroxylation, addition of water to this arginine with the help of enzyme arginase yields urea, and ornithine is formed, which goes back and continues the cycle. Now if you remember in the question, they said the child has increased amount of citrulline. So citrulline is a major component of the urea cycle, and accumulation of citrulline means that there is a defect of enzyme arginosuccinate. So arginosuccinate synthetase, correct? Arginosuccinate synthetase defect is called as Citrullineia, in which blood has increased quantity of citrulline, and this is a disorder of urea cycle. So that is why the answer is urea cycle or ornithine cycle is disturbed. Same name, same answer. Urea, ornithine, citrulline, the same thing. 160. A male patient has been diagnosed with acute radiation disease. Lab exam revealed a considerable production of platelet serotonin level. The likely cause of disturbed serotonin reduction is the disturbed metabolism of the following substance. If you remember, serotonin is produced from amino acid tryptophan. Tryptophan is converted into 5-hydroxytryptophan with the help of enzyme tryptophan hydroxylase, which is further converted into serotonin with the help of the enzyme aromatic amino acid decarboxylase. So, serotonin is directly converted from 5-hydroxytryptophan, which is the answer over here. Correct? Simple. So let's go to the next one. 161. A dietary intake of a 30-year-old nursing woman, which means she is feeding the baby, contains 1000 mg of calcium, 1300 mg of phosphorus, 20 mg of iron per day. It is necessary to change the content of these mineral substances in the following way. So the daily intake of some ions I provided. Calcium is around 800 to 1200 mg, phosphorus is around 1500 to 2000 mg, and iron is around 18 to 25 mg. So the woman here has the less amount of phosphorus intake, and I think we need to increase this phosphorus content. That is the answer over here. Pretty simple. Remember the daily intake values of these ions, or at least remember the ratio, that calcium and phosphorus have almost 1 to 2, 1:2, correct? 800 to 1600, 1200 or 10,000, 1000 to 2000, so around that way. Anyway, 162. Cardinal symptoms of primary hyperparathyroidism are osteoporosis and renal lesion, along with the development of urolithiasis. What substance makes up the basis of this caliculi in this disease? If you remember, hyperparathyroidism means that there is increased amount of calcium in the body. Yes, increased amount of calcium, because parathyroid hormone is responsible for increasing calcium levels in the blood. Now, if there is hyperparathyroidism, there would be hypercalcemia, and hypercalcemia will result in increased amount of calcium phosphate stones which can occur in the kidney. 90% of the stones that occur in the people are calcium stones. So other stones include cholesterol stones, bile stones, oxalate stones. But hyperparathyroidism leads to hypercalcemia. Calcium levels are increased in the body. An abnormal increase of something can lead to accumulation of these things in various tissues. So calcium increase leads to accumulation of calcium phosphate into the kidney, causing kidney stones, urolithiasis. Correct. 163. Study of conversion of a food coloring agent revealed the neutralization of this xenobiotic, which takes place only in one phase, microsomal oxidation. Name a component of this phase. The Cytochrome P450 family is an interesting family. It contains heme as the cofactor, and they function as monooxygenases. Now the function, first of all, the location of Cytochrome P450 is in the liver cells mainly, but they can be located in the cells of the throughout the body. Now microsomal Cytochrome P450 in the liver, what is found in the liver, P450 that is found in the liver, they are responsible for detoxification of drugs and foreign compounds by being involved in the hydroxylation reactions which involved NADPH. This is the function of Cytochrome P450. They help in detoxifying various drugs, foreign compounds which are called as xenobiotics. So this is the function of Cytochrome P450, and that is why neutralization of these xenobiotics takes place during microsomal oxidation, and the component of this microsomal oxidation, important component, is Cytochrome P450. 164. A patient had an hemorrhagic stroke. Blood examination revealed strengthened kinin concentration. The patient was prescribed Contrical. It was administered in order to inhibit the following proteinase. Now, Contrical is an anti-protein drug. It destroys the protein containing enzymes. So Kallikrein and Kinin, we need to understand what is the function of Kallikrein and Kinin. So Kallikrein and Kinin system occurs in this way, that various factors activate pre-kallikreinin to kallikreinin, and this kallikreinin converts inactive kinogens into kinins like bradykinin and kallidine. And these kinin are responsible for various functions like inflammation, during inflammation, for mucosal secretion of electrolytes, for metabolite production, for contraction of smooth muscles, for hypotension. They have vasodilation effect. So this is how kinin and kallikrein system is linked. Kallikrein activate kinogens into kinin. So that is why, because there is a increased kinin concentration, so Contrical acts to inhibit kallikrein. So if you inhibit kallikrein over here, then kinogens will not be converted into kinins, and you can inhibit the effect of these kinins by inhibiting the synthesis of kallikrein or inactivating kallikrein. So that is how Kallikrein and Kinin system works. Simple example. 165. A 49-year-old driver complains about unbearable constricting pain behind the breastbone, irritating to the neck. The pain arose 2 hours ago. Objectively, the patient condition is grave. He is pale. The heart tones are decreased. Lab studies show that there is a high concentration or high activity of creatine kinase and first lactate dehydrogenase, isoenzyme 1. What disease are these symptoms typical for? So we talked about this. Say there are certain enzymatic markers that help us to diagnose myocardial infarction. So there are these enzymes which are present in the myocardial tissue. Now when there is an injury of myocardium, what happens is that these enzymes, because of cellular injury, they are thrown into the blood, their concentration increases in the blood, and if we analyze their concentration, by analyzing the blood, we can find out that if there is an occurrence of myocardial infarction in the patient. Now, the first enzyme to get activated is creatine kinase. It activates around, say, 2 to 5 hours after the myocardial infarction. It elevates 2 to 5 hours after myocardial infarction and is there in increased amount for up to 2 days. Then aspartate aminotransferase, AST, is elevated. Then lactate dehydrogenase first and second are elevated. So remember, the enzymes which will be elevated in myocardial infarction, which are the intracellular markers, are the creatine kinase MB form. Remember, the second isoenzyme. Aspartate aminotransferase, also called as SGOT, lactate dehydrogenase first and second, which is present in the heart and RBCs. But if all of them are elevated together, it clearly shows that myocardial infarction has taken place. So always go for acute myocardial infarction if you see elevation of creatine kinase, lactate dehydrogenase. And all the other symptoms also point to, during myocardial infarction you have this pain irradiating to the neck from the sternum, the breastbone. So heart tones are also decreased, bradycardia occurs. So that is all for today. Oh, we have one more. 166. Plasmic factors of blood coagulation are exposed to post-translational modification with participation of vitamin K. It is necessary as a cofactor in the enzyme system of gamma carboxylation or protein factors of blood coagulation due to increased affinity of their molecules with calcium ions. What amino acid is carboxylated in these proteins? Vitamin K, what does it do? It is a coenzyme for carboxylation of glutamic acid. So glutamic acid is carboxylated into an inactive precursor of clotting factor like 2nd, 8th, 9th and 10th. These are the clotting factors in which vitamin K is involved and it carboxylates glutamate into their structure. What happens? There is an extra carboxylic acid present in the clotting factors due to this process, and by which they can get attached to the calcium ions which carries positive 2 charge, and now when they lose their hydrogen, one of them carries minus and the other one carries minus, so both they can get attached to a calcium ion which carries 2 plus, and they undergo the process of coagulation. So this is the mechanism in which vitamin K carboxylates glutamic acid amino acid into the precursors of clotting factors. So 167. Pharmacological effects of antidepressants are connected with inhibition of an enzyme catalyzing biogenic amines, noradrenaline and serotonin in the mitochondrion of cerebral neurons. What enzyme participates in this process? So if you remember, we did this in the last video. Monoamine oxidase is the enzyme responsible for degradation of these amine neurotransmitters. Which amine neurotransmitters? Norepinephrine, epinephrine, serotonin and dopamine. Now MAOA and MAOB are the two isoenzymes of monoamine oxidase. They use various substrates for degradation, like MAOA uses serotonin and degrades it. It degrades norepinephrine, dopamine and tyramine. MAOB degrades dopamine and phenylethylamine. And their localization is different. MAO is present in brain, MAO B is present in brain, but MAO A is also present in gut, liver, placenta and skin, and MAO B is present in platelets and lymphocytes. And in the platelets, what does it do? The platelets, they carry serotonin, correct? And when the serotonin is to be used, monoamine oxidase is responsible for their degradation. 168. An oncological patient was prescribed methotrexate. With the lapse of the time target, the cells of tumor lost susceptibility to the drug. There is a change of gene expression of the following enzyme. If you remember, we did this in the first video of biochemistry series. A quick revision: methotrexate is responsible for inhibiting the enzyme dihydrofolate reductase. This dihydrofolate reductase is involved in DNA synthesis or mononucleotide synthesis by what? Now tetrahydrofolate, a donor, donates methyl group to the enzyme of thymidylate synthetase, which is responsible for converting dihydroxy uracil monophosphate into dihydroxy thymidine monophosphate. Now what happens that after donating it gets converted into dihydrofolate and it cannot be used. For that it to be a methyl donor, dihydrofolate reductase takes a folic acid and adds it into the dihydrofolate and forms another tetrahydrofolate so that it can donate methyl group again. But if this enzyme is inhibited, which is seen by the action of methotrexate, this whole process cannot occur because there would be a scarcity of tetrahydrofolate, and more dihydrofolate will be found in the blood. So this is the mechanism of action of methotrexate, and this is used to treat cancer cells. How? By inhibiting DNA synthesis in cancer cells, it causes the death of cancer cells. So that is why the answer here is that it changes the gene expression of dihydrofolate reductase, the site of action of methotrexate. So that is all for today, and that is the end for biochemistry base. I hope you understood all the questions and I hope you liked our video series. So I don't have anything else for you right now. So all the best for your Krok, and we'll be uploading the remaining physiology base last part in 1 or 2 days. So that would be all. This has been good. See you in the next one.